My name is Sachin Mathur. I'm a computational scientist in Sanofi. And in this talk, I will go through the algorithm that we have developed to discover potential endotypes in the time series clinical transcriptomics data. So the time series data in the clinical domain has been generated for over 20 years now, and with the main objective of understanding how the dynamic systems evolved with time. And this was done mainly to find biomarkers in diseases, as well as to find gene targets. Now, when we go to NCBI and GEO and search for any kind of clinical transcriptomics data, most of the data is being generated from the bulk transcriptomics. Even though single cell has been around for about 10 years now, most of the data sets are from microarray or with bulk RNA-seq. With analyzing the transcriptomics data in the clinical domain comes with its own set of challenges. The first one is with demographic differences between patients with respect to their age, gender, prior disease history, the concomitant medications that the patients are taking. So all of these factors alter the gene expression in patients, and they can lead to differential response to drug treatment. The second thing is that the patients may be in a different disease state for a given visit. So for example, you have a set of patients, and at visit 1, the patients may actually be in slightly different disease states. So if you try to compare a set of the gene expression of patients at say a time point 1 versus time point 2, you have to contend with the intra-variability in the gene expression at a single time point. And this comparison will give you suboptimal results. The other thing is that there could be missing data points because the patients may drop out of clinical trial. And so at a given time point, you may have very few patients to contend with. And the comparison between time points may not yield very good results. The third one is the patients may be having different subtypes of disease. So for example, in breast cancer, you have different subtypes like basal-like, HER2, etc. And when you have the different subtypes of disease, some patients may respond very well to a drug, while the other patients may have, they can respond very in a partial manner or not respond at all to the drug. A case in point was the recent pandemic from COVID-19, where people from the same household, some of them had a very severe form of the disease and had to be hospitalized, while others remained asymptomatic or had a mild form in which they were able to recover within a week. So this points to that there could be different subtypes of the disease among patients who are sharing the same diet and having same environmental factors. So to tackle this problem, there have been many methods, and they actually form into two paradigms. The first one is based on the expectation maximization-based methods. The aim here is to reorder the samples so that there are smooth changes in gene expression. And this is usually ordered by using polynomial curves of second or third degree. The underlying assumption here is that there are smooth gene expression changes with respect to disease progression or with treatment response. And the aim is to reorder the sample so that we get smooth gene expression. The main limitation here is that the clinical data would be very sparse. For example, if there are very few patients, for the polynomial gene curves, you have very few support points. So they don't fit very well. The other class of methods is based on the trajectory inference methods from the single cell data. In this, the objective is to order these patients so that again, going back to the single cell, the main goal is to order these cells, and the gene expression similarity between cells is based on the assumption that they are coming from a similar or the same lineage. So if we imagine a path along the gene expression space in single cell, it's basically maximizing the cell-to-cell similarity. So extrapolating those methods to the samples here, It's basically we are trying to maximize the sample to sample similarity in the gene expression space. The main limitation of this approach is that in single cell, you have thousands of cells, but you don't have that luxury in the bulk RNA-seq. For the trajectory construction, there are multiple algorithms, but most of them are, the most popular is the minimum spanning tree, or you have the information-based methods as well. Another constraint, another limitation of this method is that they are not time aware. That means the samples that are collected at a later time point can actually come before the prior time points. However, a couple of exceptions exist, namely the tempora and supper time. So in light of these limitations, we have developed a new algorithm called Truffle, which stands for trajectory inference via multi-commodity flow with node constraints. It performs through-time ordering of samples in short time series data, and by using these trajectories, we can infer endotypes. So we have actually used an existing algorithm called multi-commodity flow and adapted it to the time series data. In the multi-commodity flow algorithm, which is actually based in operations research, the aim is to transport a set of commodities like oil from source to destination, and the source and destination are connected with a network of highways. Each is associated with a cost and demand. The aim is to transport these commodities in such a way that the time is we are minimizing the time as well as minimizing the cost. So now adapting this algorithm from operations research to time series data, first we construct a graph. And the way we start is for each sample, we find the nearest neighbors using KNN or UMAP, and we construct a graph. And then, for example, if you have a patient in red, and the patient has 3 time points, T0, T1, and T2. We start with the initial time point at T0, we find out the nearest neighbor. And this nearest neighbor could be from the same patient, or it could be a sample from a different patient. And by using these transitions to the nearest neighbor, we construct a trajectory for that particular patient. There are 3 constraints in this graph problem. The first one is the maximum edge capacity, the maximum node capacity, and the flow conservation. Maximum edge capacity basically ensures that we are not using one edge in the graph too many number of times. Maximum node capacity ensures that a node is not used more than n number of times. And basically those 2 constraints, we make sure that we are reducing the noise and reducing the effect of an outlier sample. Flow conservation basically ensures that for a single patient, only one trajectory is constructed. The objective function over here is the utilization of an edge times the capacity of the edge. The capacity of the edge, or the cost of the edge, is basically the Euclidean distance of the PC embeddings of the samples. So we minimize this cost, the objective function, by using these 3 constraints and produce a trajectory for every patient. It is important to note that we try to find out the nearest neighbor for a particular sample. And if you think about the clinical trial, it usually has about 3 to 4 time points, and you're getting snapshot of the disease state for a patient, and you get only about 3 to 4 snapshots, which is not the complete trajectory of the disease for a patient. So we make use of the disease states of the other patients by using these samples, and we allow the transitions from the patient sample to go to samples from other patients, basically filling in the blanks for the disease state for a particular patient. So in this example, we have the initial time point as T0, and we transition it to a black node here. That means it's a sample from another patient, and then we make sure that it is the final node is the final sample of that particular patient. And that is how we construct the trajectory for a given patient. Independently of that, we also cluster these samples of all these patients together by using a clustering algorithm called Leiden, and extract the genes associated, extract the genes from each cluster, compare them to the control samples, and get the differentially expressed genes and pass them to GSEA to get the biological mechanisms that are enriched in a particular cluster. The aim of doing it is to extract the disease states from each of these, from all of these samples in the patients. And in the final step, we actually combine the two. We take the trajectory of a patient and then impose the disease states of the extracted disease states. So for example, here we have a patient in red passing through about 6 nodes, and the 6 nodes can be colored as 2 in green, then orange, and then the 3 in yellow, leading to a trajectory of green to red to yellow. So this is inferred as a trajectory of disease states for that particular patient. We make sure that the timing considerations are met. In the sense, we start at the initial time point of the initial sample of the patient, and then we terminate at the final sample for that particular patient. And given that we are making these smooth transitions, we are capturing the intermediate disease states of that particular patient, even though we don't have samples for that particular patient. We ensure that we ensure that the trajectories are smooth. And once we have all the trajectories for the patients, we basically stack them up and find out the most important trajectories or the most common trajectories between patients, and we refer to them as endotypes. We validated this method on 3 data sets: the Crohn's, COVID-19, and psoriasis. Crohn's and psoriasis they were influential studies. They had the patients were treated with a biological, whereas the COVID-19 was an observational study. And here are the results from the Crohn's disease. On the far left, you see the multi-commodity flow graph, in which each of these nodes are colored with disease state. And once we get this disease state, in the disease state, we have clustered samples. And the next step, we take the trajectory, the inferred trajectory, and extract the genes and put them through a clustering algorithm called STEM, which is the short time series expression minor. And using STEM, we find genes which are similarly regulated across time. So one of these results is that we find a bunch of genes which are downregulated, which is involved in immune response. And from prior literature search, we know that these patients are actually responding to the treatment. In the other trajectory, we see a bunch of genes which are going up with time, and these patients are not responding to the drug. So from the same data set, we find 2 different trajectories, one set which is responding to the drug and the other one not responding to the drug. For COVID-19, we observed 3 trajectories. In the first one, the genes responsible for immune response, they go up in the initial time point and then they go down. We also see a similar behavior in the sensory perception of smell, where the genes are, again, going up initially and then they go down. From prior literature searches, we know that the sensory perception of smell is extremely important, and we are not really sure as to why they go up, which is a positive signal, but they go down. In the second trajectory, we see that there's a monotonic increase of sensory perception of smell, while there is steady downregulation of the immune response, which is indicative that the patients are progressively getting better with time. And in the third, we see that the the genes belonging to an immune response are gradually going down, but the sensory perception of smell is not detected. So from this COVID-19 data sets, we detect about 3 endotypes. So to compare with earlier methods, the other methods, we used 2 methods. One is supper time. It's basically extract genes by using a logistic regression, which explain the transition from one time point to the other. We applied Go analysis on that, and we obtained only a 1 Go process, which we couldn't explain it in the context of COVID-19. The second method we compared against is called Tempora. Tempora is a method which uses not only the gene expression, but also the pathway information to construct trajectories, and it uses mutual information to construct those trajectories across different patients. In Tempora, when we use these the trajectories and extracted the gene expression profiles and sent it through STEM, what we found is many of these gene profiles were oscillating with time. So it was kind of confusing to understand as to what was happening from the biological perspective. It did detect the sensory perception of smell, but again, the trajectory was oscillating, so we couldn't gauge whether the patients were improving or not. So to conclude, we have developed a method that can integrate data from time series on heterogeneous individuals. It overcomes issues related to response rate and sample times while allowing for multiple endotypes representing different trajectories. We validated Truffle on 3 data sets, and we were able to successfully uncover the underlying endotypes. We have tested the algorithm, and we have applied the algorithm on a data set which had about 600 samples, and it took us a few minutes to run. But if we extrapolate it to more than 1,000 samples, we suspect that it's going to take quite a bit of time because this problem is NP hard. So we'd like to thank the reviewers for the valuable suggestions, which led to the improvement of the manuscript. We'd like to also thank Sachin Mathur from Precision Medicine Computational Biology Group for suggesting us the Crohn's disease dataset. And this work was a result of a summer internship last year. We have positions open for the summer 2025, and they should be posted on the careers website in December. So thank you very much.
Thank you.
Yeah, very nice talk. Full disclaimer, I'm a computational biology of Takeda. So I'm wondering, when you impute your data, like you do this patient by patient, subject-wise, right? But numerically, how did you deal with the transcriptome data? Are you talking about single-cell RNA-seq or just like bulk RNA-seq? And did you have any special consideration for each individual gene feature? I expect you will publish. I definitely will learn more. But do you happen to have any quick comments and feel comfortable to share?
Yeah. So the data is all publicly available, and this is done on the bulk RNA-seq. And for each sample, we use all the genes. So basically, we are transitioning from one sample to the other, and we are calculating the Euclidean distance among the PC embeddings for those samples. So we are using all the genes.
Okay, very nice. So another quick question. I think when you do the imputation step, you don't consider any endotype information.
No, we do not consider any kind of endotype information. And imputation is basically the transition from one sample to the other.
Yeah.
Right.
But did you consider, for example, you have T0 time baseline and T1 and T2, you consider the prior time points, but do you consider any time points afterwards, after the one you need to impute?
We start with the initial time point for that particular patient, and then we transition through the graph by using the nearest neighbor. And we make sure that all the samples for that particular patient are covered, and it terminates at the final time point for that particular patient.
Hi, thanks for the talk. I was wondering if you consider the patient attributes when you're trying to predict the trajectory for, like, say, patient one, because we know age and sex have an importance on disease trajectory.
That's a good question. And currently, we do not do that, but it can be used as additional covariates. Yeah.
Thank you for a very interesting talk. Hey, I was wondering whether you could apply this method also to other kinds of data, because the problem that you're trying to address is a very common problem that's not only important for gene expression, but also, for example, for clinical data or all kinds of other like biological data sets.
Yes, if we have clinical data, again, with the gene expression, you understand the biological system, but yeah, it can be used for, the algorithm is quite generic, right? And it can be used for clinical covariates as well.
Thank you.
Yeah, thank you for the very interesting talk. I think this is a really useful tool, for us to study this disease dynamics. So I just one quick question: when you make the trajectory inference, right? You mentioned you use all genes. I know this is an unbiased method, but for specific disease, not all genes are equally important, right? So did you try to do some sort of feature selection, or maybe use prior knowledge to pre-select sort of most critical genes, and that use that subset of genes to inform the trajectory might better face the disease more specifically?
Right. So we did perform gene filtering with respect to their counts in the RNA-seq, bulk RNA-seq. But for most part, we used a large gene set. However, we are comparing these genes to the control expression. So when we use all genes with control expression, and then we feed that into GSEA. So even though all the genes are not important, many of that is getting filtered in the GSEA state.
Yeah. Okay. All right, thank you.
You're welcome.
Thank you, and let's thank